I originally started my research studying genes, which we know the human genome is what dictates how our cells function. It turns out that every single gene within the human genome codes for a protein, and the protein is the actual workhorse of the cell. What's emerging over the last 50 years is that every single protein is modified in various ways on various parts of its structure by what we call post-translational modifications, or PTMs. And these PTMs can change how a protein functions, how it does its job, and therefore how a cell responds to its environment. We are trying to answer a number of questions surrounding the idea of PTMs, or post-translational modifications. And one of the major questions is, can we predict whether or not a specific PTM on a protein will have any effect on biological function of that protein? So we've merged with the bioinformatics program here in the School of Biology, and with a master's student from that program, we've developed a tool that allows us to predict function of any modification on any protein based on the structure, the three-dimensional structure of that protein. The most important and surprising finding so far has been use of this bioinformatics tool that we call SPOP, or structural projection of PTMs, to interrogate a very specific protein complex called the heterotrimeric G protein complex. This is a protein complex, meaning it has multiple proteins that are stuck together, and together these have a specific function. They help the cell respond to extracellular stimuli like light or hormones or neurotransmitters. And so we've used a bioinformatics tool that we've built to look at how heavily modified this protein complex is, and to try to determine whether or not there are certain parts of the structure of the complex that are more heavily regulated by modifications than others, and then to figure out why that's the case.